Cryo-Fluorescence Tomography, or CFT, is a relatively new imaging technique, and in this video we describe how it can be applied to colorectal cancer research using the Xerra system. The rainbow colon mouse uses green fluorescent protein, or GFP, which is co-expressing with PI3 kinase upregulation, in order to visualize cells that are transitioning from adenoma to carcinoma. The CFT technique involves freezing the mouse in a white block of OCT compound, as seen here. After exposing a surface of the tissue, we illuminate with red, blue, green, or infrared lasers while acquiring data off of the block face. The blade then moves across the block face, cutting a thin layer, 20 to 50 microns thick, off the surface, and the whole process can repeat hundreds or thousands times. In this way, we build up a stack of white light and fluorescent images. This fly-through video is showing us slice-by-slice images as they were acquired off the block and superimposed onto each other. Green and red fluorescent channels can be seen overlaying on the color anatomical images as we scroll through the whole volume of the mouse. We are now showing fly-through videos of the separate fluorescent channels. The GFP expressing cancerous tissue on the left shows us several large primary tumors, and we see many smaller tumor clusters throughout the gastrointestinal tract. On the right is the normal tissue that is expressing TD tomato. The stack of images is reconstructed into 3D, and we now see a high-resolution tomographic fluorescent image of the tumors in green. The 3D dataset is one of the whole mouse in normal tissue in the red. Each voxel is a ratio of adenoma to carcinoma. So with this data, you can easily see the tumor progression by the number and size and distribution of the tumors and the organs that are affected by this cancer. This slide shows all of the data together. From the left are 4 coronal fly-throughs off the block face of white light, GFP, and TD tomato images and an overlay. On the right, we see the tomographic GFP and TD tomato maximum intensity projections. Researchers can now use this complete picture of colorectal cancer to develop new therapies and quantify the efficacy of these therapies. Without Xerra, the researcher would have to dissect all of the GI tract, flush, cut into smaller volumes, mount on a cryostat block, slice, mount onto slides, stain, and image. This extremely labor-intensive method is expensive and time-consuming and only lets the researcher sample a small fraction of the tumor tissue. For more information, please contact us at emitimaging.com.